Chapter 4, Topic 4.2, Structures of the Eukaryotic Cell. In this topic, we will be discussing the structures that most eukaryotic cells have, including external structures such as cilia, flagella, and the glycocalyx, boundary structures such as the cell wall and the cytoplasmic membrane, and internal structures, which we're going to be going over in more detail. At the end of this recorded lecture, you should be able to differentiate between cilia and flagella in eukaryotes, and between flagella and prokaryotes and eukaryotes, list which eukaryotic microorganisms have a cell wall, list similarities and differences between eukaryotic and prokaryotic cytoplasmic membranes, describe the important parts of a nucleus, diagram how the nucleus, endoplasmic reticulum, and Golgi apparatus act together, explain the function of the mitochondria, explain the importance of ribosomes and differentiate between eukaryotic and prokaryotic types, and list and describe three main fibers of the cytoskeleton. Under the heading of external structures, we're going to talk about cilia and flagella and the glycocalyx. Eukaryotes have structures for motility or moving the cell through the environment. In multicellular organisms, one of these structures, cilia, have been adapted to moving materials in the environment past the cell and its tissue. But in this class, we're going to be focusing on unicellular organisms. Flagella in eukaryotes have a very different structure than prokaryotic flagella. Eukaryotic flagella are composed of bundles of microtubules that are arranged in a 9+2 arrangement, like in this micrograph. 9 bundles of 2 microtubules are arranged in a circle around the core. ATP is used to pull the bundles one way or the other. This results in a back and forth motion or rowing motion. I have a video of a sperm cell that shows the motion of eukaryotic flagella very well, but I'm also going to show you a video of a prokaryote, Salmonella, that shows the difference in the movement between prokaryotes and eukaryotes. Here you see some bacteria swimming around. You'll notice the flagella go 360. They're like little outboard props. See, it's going around and around and around. Here we've got a human sperm, and there's a flagellum coming out of the back. And you'll notice that it goes back and forth in a wave motion, as opposed to with the bacteria, where you had it turning 360 like a little propeller prop. So that is the difference between the motion of bacterial flagella and eukaryotic flagella. Cilia are composed of the same 9+2 arrangement of microtubules. They're basically short flagella. And organisms that have cilia usually have a lot, like in this example. Now I've got a video of some lovely cilia movement, and we'll take a look at that. Now if you look down here in the lower corner, you can see some really nice cilia movement. And see how they move in a coordinated fashion? It kind of reminds one of spectators doing the wave. Next up is the glycocalyx. Like prokaryotes, eukaryotes often have a glycocalyx. It is usually composed of polysaccharides. In multicellular organisms, it can be called the extracellular matrix. In any case, it functions in protection, attachment to other cells and to surfaces, and in communicating with other cells. Sounds like the glycocalyx in prokaryotes, huh? Under the category of the boundary structures, we're going to talk about the cell wall and the cytoplasmic membrane. Some eukaryotic cells have a cell wall. Fungi have a cell wall that is composed of chitin, and algae have cell walls composed of cellulose. The text says that some fungi have cell walls composed of cellulose, but any fungi that had cell walls composed of cellulose got kicked out of the fungi and into the algae. Animal and protozoa never have cell walls. The cytoplasmic membrane of eukaryotes is composed mostly of phospholipids in a bilayer, just like in bacteria. Eukaryotic cytoplasmic membranes also have sterols, which are 4-ringed lipids. Cholesterol is an example of a sterol found in animal cell membranes. They provide stability to the cytoplasmic membrane, which is vital for those cells that don't have a cell wall. Cytoplasmic membranes are selectively permeable in both eukaryotes and prokaryotes. And in both eukaryotes and prokaryotes, they have transmembrane proteins for moving in the big stuff. It's just there are different transmembrane proteins in prokaryotes than there are in eukaryotes. On to internal structures. We're going to be talking about the nucleus, the endoplasmic reticulum, or ER, Golgi apparatus, lysosomes, vacuoles, mitochondria, chloroplasts, ribosomes, and cytoskeleton. As we have often said, eukaryotes are defined by having their DNA enclosed in a membrane. This organelle is called the nucleus. The nucleus has a double membrane with pores. See, here's a big blow up of the pores that allow protein and enzymes to be brought into the nucleus and for RNA and other molecules to leave the nucleus. Inside the nucleus is a structure called the nucleolus. This is the site of ribosomal RNA, or rRNA, synthesis. The subunits of ribosomes are assembled there before they are transported back out into the cytoplasm. The nucleus is connected to another membranous structure called the endoplasmic reticulum, or the ER. This is the site for enzymes that synthesize various molecules within the cell. If it is studded with ribosomes and it makes proteins that will be embedded in a membrane, it's called the rough ER because it appears rough in an electron micrograph. Some mRNAs from the nucleus come directly into the rough ER to make the proteins. If the ER is not studded with ribosomes, it is studded with enzymes that are not easily visible under an electron micrograph. These enzymes synthesize lipids and carbohydrates for the cell. This ER is called the smooth ER because it appears smooth under microscopes. Little blebs of membrane, or vesicles, will pinch off from the ER carrying proteins and lipids and carbohydrates to the Golgi apparatus. The Golgi has enzymes for modifying macromolecules that it gets from the ER. It may combine a lipid and a carbohydrate to make a liposaccharide, or it may combine a lipid and a protein to make a lipoprotein. It may modify the macromolecules in other ways too. You can think of the ER as the factory that makes the parts for the cell, say sheet metal and bolts and nuts. Using that analogy, the Golgi would be the factory that takes the parts to make the finished product, say a car, from the sheet metal and the nuts and the bolts. Vesicles then pinch off from the Golgi and take the finished molecules to where they need to go. The nucleus, ER, and Golgi all work together to manufacture what the cell needs. One of the vesicles that can pinch off from the Golgi is a specialized vesicle called a lysosome. It contains hydrolytic or digestive enzymes that are made in the rough ER and modified in the Golgi. Then they are pinched off into the lysosome, and being wrapped in a lipid membrane protects the rest of the cell from their action. Lysosomes can fuse with other vacuoles, and these vacuoles can contain a lot of different substances, but let's say that one of them contains food to be digested. The enzymes in the lysosome can combine with the food vacuole and fuse, and the hydrolytic enzymes will do the job of breaking down the food without destroying the ER or other important structures in the cell. Vacuoles can also contain wastes or enzymes for export out of the cell, or products that need to go to another part of the cell. Think of them as the UPS trucks of the cell. Mitochondria are the energy producers for eukaryotic cells. They have two membranes. The outer membrane is smooth and marks the outer boundary of the mitochondrium. The inner membrane is folded and is the site for enzyme complexes used in aerobic respiration. We'll talk about them more later. Mitochondria have their own DNA and 70S ribosomes, which is evidence that they were once free-living prokaryotes that have adapted to a symbiotic life within the eukaryotic cell. They also divide independently of the rest of the cell. Chloroplasts are similar to mitochondria in that they have two membranes, There's the outer one, there's the inner one that has lots of little folds. Their own circular DNA, 70S ribosomes, and they also divide independently of the cell they are in. Chloroplasts function in oxygen-producing photosynthesis, and they are found in algae, plants, and some protozoa. Cells that have chloroplasts always have mitochondria too. Ribosomes in eukaryotes are 80S, which is bigger and a different shape than prokaryotic ribosomes. Here we can see the small unit is a 40S, the large subunit is a 60S, and they combine to make an 80S ribosome. Ribosomes make all the protein in the cell, and they are composed of proteins and rRNA. Ribosomes can be found bound to the rough ER or floating, so to speak, in the cytoplasm. Crisscrossing the cytoplasm of eukaryotic cells is the cytoskeleton. Just like the name sounds, the cytoskeleton can give support and shape to the cell, like the human skeleton does to the human body. Unlike the human skeleton, the cytoskeleton can also be involved in changing the shape of the cell, transporting organelles and molecules within the cell, and so on. The cytoskeleton is composed of 3 types of fibers: actin filaments, intermediate filaments, and microtubules. Actin filaments are the smallest of the cytoskeleton fibers. They are found in highest concentration under the cytoplasmic membrane. They are involved with cell division, contraction of the cell, cell extensions, and pinching off of vacuoles and lysosomes. Intermediate filaments are medium-sized fibers. Their main role is structural reinforcement of the cell and of organelles, such as the nucleus. In multicellular organisms, they are involved in cell-to-cell connections. Microtubules, right here, are the largest fibers. They form the spindle that separates chromosomes during cell division. They form the internal structure of flagella and cilia, and they help maintain the shape of cells that don't have a cell wall. In summary, I wanted to show you a diagram that shows everything that we've gone over. Make sure you know all the structures and their functions, and pick them out on this or another diagram of a eukaryotic cell. Well, that's it for this topic. You should be fairly comfortable with the structure of a eukaryotic cell, being able to differentiate between cilia and flagella in eukaryotes, and between flagella and prokaryotes in eukaryotes. List which eukaryotic microorganisms have a cell wall. List similarities and differences between eukaryotic and prokaryotic cytoplasmic membranes. Describe the important parts of a nucleus. Describe how the nucleus, endoplasmic reticulum, and Golgi apparatus act together. Explain the function of the mitochondria. Explain the importance of ribosomes and differentiate between eukaryotic and prokaryotic types. And list and describe 3 main fibers of the cytoskeleton.